with that, I'd like to hand off to Professor Angela Belcher to introduce our speaker.
Well, thank you, Justin. And this lineup looks fantastic. It's going to be a great summer in the biomaker space and learning about all the contributions here and around the Boston area in development of tools and technology and science around COVID. So thank you so much for organizing this. So I have the pleasure of introducing Professor Mike Yaffe, our speaker today. And to go through his CV would take the whole time, so I'm just going to say a few words about him. He's the David H. Koch Professor of Science, and he's the director of MIT's Center for Precision Cancer Medicine. And probably most important to me, he's a member of my faculty in biological engineering and also of the Department of Biology. Somehow between those roles, he's also manages to be a full-time active intensive and neuro intensivist and attending surgeon at Beth Israel Deaconess Medical Center. And probably, again, most important to me, he's just a fantastic colleague. You'll never meet a more enthusiastic or passionate scientist or engineer and teacher. He's an extraordinary teacher and mentor as well, and just all around great colleague. So for me, it's really fun to get this opportunity to introduce him today. I should point out also that he's a retired colonel in the United States Army Reserve Medical Corps, where he's been very active and deployed overseas, attending our troops, which is amazing as well. I'll just take a few more seconds because he has so many important things to say. He's an engineer. His BS is in material science and engineering from Cornell. His PhD is in biophysical chemistry and pharmacology from Case Western. His MD is also from Case Western, and I think I said his PhD was from Case Western. And then he has a lot of training at various hospitals, which I won't go into detail. He has many awards, teaching awards, which are extremely valued here at campus on MIT. He also has a Bronze Star Medal for service in Afghanistan, and his research was cited for a signaling breakthrough of the year by Science magazine. So I'm not going to take any more of this valuable time, but to say welcome, and I can't wait to hear about what you're doing today.
Thank you very much, Professor Belcher. That was a very kind introduction. I often people ask sometimes, why I do all these things, and I often joke, as I did when my mother was still alive, that one of these days I'll figure out what it is I want to be when I grow up. I'm just not there quite yet. So with that, I'm going to share my screen, and hopefully this will work. And if I've done this correctly, and someone's going to have to unmute and know if this works, you should be seeing a PowerPoint slide that says STARS trial. Justin, can you see that?
Yep, you look good.
Okay. So what Justin asked me to tell you a little bit about is an idea that came actually something that do with COVID therapy. And in the process, I'll try to tell you where I think there's a lot of avenues for involvement from people in the biomaker space. So this trial is collectively called the STARS trial, and it's the study of alteplase and respiratory failure. And alteplase is just a molecule called TPA, or tissue plasminogen activator. And I'm going to go through really the scientific rationale of why we started the trial. And that really starts with this slide. So this is an X-ray. And can people see a pointer when I move my pointer on the screen? Okay, so this is a classic X-ray with COVID-19. And this white thing in the center is, of course, the heart. And what the sides should be the same color black as what you see up here, but it's not. It looks very whitish, and that kind of whiteout, or so-called ground glass appearance, is sort of the hallmark of the lung failure that we're seeing in patients with COVID-19. And although this X-ray looks a lot like many other types of lung failure that we see in the intensive care unit that's caused by a lot of different things, there's something very funny about this type of lung failure. It is very different than the lung failure that we have seen until COVID-19, despite the fact that the X-rays look pretty similar. And what's different is the mechanical properties of the lung. If you measure the elasticity of the lung, right? Like a stress strain, which in this case, the stress would be the airway pressure and the strain would be the amount of distension of the lung, what you would find is that the elasticity of this lung is relatively normal, despite the fact that it looks so terrible. Now, an interesting thing about the patients who have these types of lungs is they aren't aware of how bad their oxygenation is and how poor their lung is functioning. And that's because one of the things that you sense when you feel like you're short of breath is you sense that decreased elasticity, that decrease in Young's modulus of your lung. But if your lung is normally elastic, you won't be aware that you're having trouble breathing. And the surprising thing has been people come in and they don't feel particularly short of breath because their Young's modulus of the lung is normal, but their oxygen in their blood is at a very low level. Now, this slide is meant to illustrate what we see. In this early stage shown in the blue half of the arrow up here is the initial stages of the viral infection. What I'm showing you is closer to this red stage of the lung. And the thing that is so impressive is when we see them, when the patients are finally at the point where they go to the emergency room because something isn't right, the amount of time it takes for them to present and say something isn't right until they're in complete respiratory failure, intubated and on a ventilator, is sometimes as short as 30 minutes to an hour. Something is just not right. And one of the things, as I'll talk about more, is the observation that many of these patients turn out to have a problem with the blood vessels to the lung. And they have blood clots in the vessels that deliver oxygen to the lung. And that might be a reasonable explanation about why the elasticity of their lung is okay, but they're not transferring oxygen from their lung tissue into the bloodstream because it's blocked. And this led a number of us to propose that maybe there was a role for tissue plasminogen activator as a novel treatment. And I'll explain where that comes from as we go along. Now, I'm going to show some slides, some of which are a little gross, and I apologize. But this is an autopsy specimen of what the lungs look like. Now, if I were to show you a normal lung, it would be a pale gray color. It would not look, this looks like a piece of liver, more than it looks like a piece of lung. And that's all the weight and congestion of the fluid and blood that's trapped in the lung. And some of the findings that are described by the words that you see here, in particular, I want to focus on this one, microthrombosis. That means tiny blood clots in the vessels of the lung and wall thickening of the blood vessels. Now, lest you think that this is a problem that is unique to one part of the country, this is what was described in China. This is what's seen in Italy. And what you see in these slides, if we look at the one that's labeled A up here, is this is normal, this should be normal lung. And it should have a thin, lacy appearance like in panel E. But you see it's quite congested. And all of the regions I'm showing you here, this is actually the airways that are filled with blood. And everything I'm showing you here are the blood vessels where you can see they're very congested with red blood cells. In fact, the group in Italy decided to call this disease microclots for lung vessel obstructive thromboinflammatory syndrome. This is what they're seeing in Brazil. Again, this is the lung, and what you see here is a blood vessel that's plugged with a blood clot, another vessel plugged with a blood clot, and another vessel plugged with a blood clot. This is in Brazil. And in Brazil, they describe this as fibrinous microthrombi in small-sized pulmonary arterioles. This is what we're seeing here in New Orleans. Again, notice that these lungs that you see here, these lungs are very red and very heavy. This looks, again, more like liver. And what this arrow in panel C is meant to show you is the blood clot that's sitting in one of the main vessels that perfuses this lung. This open area here is the bronchi, that's where the air goes, but the blood vessels themselves are blocked. And this is what came out of New Orleans. More recently, there was a paper that came out in Lancet that looked specifically at the lining cells of the blood vessels. And what they noticed was viral inclusions, that's shown here in panel A, and cell death, that's shown here in this inset over here, suggesting that in fact there was an endotheliitis. The endothelial cells that line the blood vessels were themselves infected by COVID-19, and this was causing inflammation in the blood vessels that was leading to blood clots. Now, this again was most recently described just a week or two ago in this paper from the New England Journal of Medicine, which described pulmonary vascular endotheliitis, or endothelial infection in the pulmonary vasculature, together with thrombosis, that means blood clots, and angiogenesis, that means the development of new blood vessels. And you can see again in this sample of lung, which should be very open and aerated, you see a lot of collapsed alveoli, the collapse of the lung, and all of these blood vessels that look like they're filled with red blood cells because they're clotted. And so our argument behind this was maybe the problem, a central problem in at least a subset of patients, are these blood clots. And if we had a way of clearing the blood clots and keeping them from reaccumulating, we might have a way that would be useful to treat these patients. Now, what's unique, as I said, about this disease is that the problem that we're seeing is not so much the virus. It really represents, shown by this orange triangle, the host inflammatory response. That is, the virus itself causes things like fever and a cough, and some alterations in some lab tests. But the thrombosis and the shortness of breath is really because of the response of your body to the virus rather than damage that the virus itself is doing, although the virus is doing some damage. What we're seeing is a new phenomenon, a phenomenon that we really haven't appreciated as much before, but that's been described in some other cases, and that's this idea of immunothrombosis. Your body's own immune reaction to the tissue damage caused in the lining cells of the blood vessels is what's responsible for then activating the blood clotting system. So that there's feedback between the normal innate immune response, things caused by neutrophils and macrophages, and the immune system, and it's reacting to this damage to the blood vessels to cause those blood clots. Now, we were interested in this, not because we had a big interest in COVID-19, but because one part of my lab has worked for many years on this with an interest on how the innate immune response, neutrophils and macrophages, might control blood clotting in situations of tissue damage, such as people that are shot, for example, in military conflicts in Afghanistan. And so when we saw this link between blood clotting and innate immunity that we were interested in trauma, in a setting of inflammation, we wondered if there might be a really important role for it. Now, I want to spend just a few moments talking about this aspect of the work, because here's a place where there's a lot of room for advancements, particularly advancements that people in the biomaker space might be able to make. When we teach you blood clotting, and some of you maybe have had it taught to you from me in 705, we teach it to you as a linear cascade, an amplifier, if you will, of proteases that starts with some damage up high and ends up making big blood clots. And the lab tests that we use are based on that concept. What we do in the lab is we take blood from someone, we spin the cells out, we take the plasma, and we add one or two things, and we measure how long it takes that plasma to make a blood clot. The problem with that test, as we now know, is it's basically meaningless for predicting what's happening in real people. And that's because in real people, blood doesn't clot by isolated plasma in a tube. It clots through the interaction of things like damaged endothelial cells that contain something called tissue factor and platelets. both of which are not present in the lab tests that we use in the hospital all the time. And this has been one of the barriers in trying to make breakthroughs in the treatment of ARDS and COVID, is that the lab tests will tell you that the blood is resistant to clotting. It's not clotting normally, it's clotting too slowly. But in the patients, the blood is clotting too quickly. And so many people have been fooled by these abnormal lab tests into thinking that there's a problem with the blood clotting being too slow, but in fact, the clotting is too quick. We know now that in fact, it's 3 different cascades. There's an initiation pathway that happens on those damaged endothelial cells. That generates a small amount of thrombin, a critical protease. But that amount of thrombin, if it exceeds a threshold, now starts an amplification process that happens on the surface of platelets, and that then propagates to give rise to a blood clot. This is an area that needs better lab tests. It's an area that needs better mathematical models, and it's an area where we don't know how to link what we see clinically with what the lab tests tell us. Now, what's missing in all of this is the link to the immunology so far, and it turns out that IL-6, one of the hallmarks of upregulated cytokines in COVID-19, controls both factors that are involved in the initiation and factors that are involved in the amplification and propagation. So IL-6 is both increasing tissue factor and it's increasing the substrates that make a clot, but it's also inactivating proteins whose normal job is to limit the extent of clot formation. And here I have to tell you for a moment about what limits blood clot formation. Now, if all we had was an amplifier, that is, once we started a clot, we amplified it and propagated it, then as soon as you got a cut on your finger, every blood vessel in your body would become a gigantic clot through this amplification process. And so there must be a balance between activating clotting and inhibiting clotting, making clots and breaking them down. And normally your body uses a terrific system to make that happen. That is, in addition to using thrombin to make blood clots out of a protein called fibrinogen, which gets turned into fibrin. Fibrin is the meshwork that makes a blood clot. There's a circulating protein whose job is to break down those fibrin clots, and that protein is called plasmin. If you had too much fibrin, you'd have too much clotting, and if you had too much plasmin, you wouldn't be able to make a stable blood clot. And so the way the system is designed is plasmin is kept inactive as an inactive form called plasminogen. And plasminogen is only activated when you have a big blood clot, because plasminogen binds to one part of the clot, and an activator of plasminogen that converts plasminogen to plasmin binds to the same clot. That's called TPA. TPA for tissue plasminogen activator. When plasminogen and TPA are both present and they bind to the surface of a blood clot, TPA will activate plasminogen to plasmin, and plasmin will then degrade that blood clot. Now, normally, again, it's a complicated process because it's inhibited, because there are things whose job is to prevent plasmin from breaking down the clot. But I'm going to skip that, but more importantly, there's also something that's job is to limit the amount of TPA, and that's called PI1. Plasminogen activator inhibitor 1. So boy, it's confusing, isn't it? You've got plasminogen activated by tissue plasminogen activator, and tissue plasminogen activator limited by plasminogen activator inhibitor. So what's the problem with this feedback loop? Well, the problem is that I'm going to jump to this slide. This is a gene expression study that came out a week ago and showed what things were upregulated in COVID-19 lungs compared to lungs that had a regular influenza response. And one of them is right here. It's called SERPINE1. And SERPINE1 is just another name for PI1, for plasminogen activator inhibitor. And so the problem you can see is that not only is there, I'm going to jump to this slide, not only is the amount of TPA that's present reduced because we have increased amounts of IL-6, but that PI1 complex, that SERPINE1, is very high. And that means that the red bars, which are the amount of normal active TPA, are very low. And so in COVID-19, we reduce the ability to make TPA, and the TPA that we make gets tied up to a normal inhibitor. And that means that once you start to make a blood clot in your lungs, you have lost the ability to lyse it. And so we reasoned if this was the case, maybe all we had to do was give people a little extra TPA, enough TPA to overwhelm that Serpin E1 that was upregulated in their lungs, and they could lyse those blood clots in their lungs. And that's the proposal that we put out. I'm going to show you some results from one patient, and then I'll tell you about the trial that's ongoing. What you're looking at here is the oxygenation in one patient's blood and the amount of adrenaline that we have to give this patient to maintain their blood pressure. And this shows you what happened within 4 hours of giving this patient one dose of TPA. I'd like you to notice that the oxygenation within a short period of time after giving TPA, the oxygenation in his blood doubled, and the amount of adrenaline that we had to give this patient to keep him alive basically got cut in half. And that shows that at least in one patient, simply activating his endogenous pathways that allowed the blood clots in his lung to lyse were sufficient to ease his oxygen carrying capability of his lung and the amount of support we had to give him to keep him alive. And so this led to the idea behind our trial. Maybe if we just give TPA to patients with COVID-19 associated respiratory failure, their gas exchange will get better, and we will reduce the amount of microthrombi in their lungs. Now, there's a lot of challenge to doing a study. You might say, well, this makes sense. Why don't you just do it? The problem is that TPA is not such a benign drug, and people sometimes bleed when they get TPA, particularly if they've had a stroke or they've had some other cause for bleeding. And so we had to design a trial that we could rapidly assess the ability of patients to benefit from, and that was amenable to change as we go. Now, one of the things you probably can tell from MIT's response to the COVID-19 crisis has been, we're trying to figure out how to do it while the crisis is ongoing. That's like building an airplane while you're flying it. So that came with this clinical trial. We designed a trial, I'll just go through the basics of it, that we can make some decisions about it in just 10 patients. We can give 5 patients TPA, not give 5 patients TPA, see if it works, figure out if we should give a higher dose of TPA, double the dose, compare that to what we saw here, and then figure out if we compare the low dose to the high dose, which dose is better. We think we'll know within 30 patients whether this is beneficial or not. As I said, there's some absolute criteria that we have to exclude. You wouldn't give this to someone with active bleeding. You wouldn't want to give this to someone who had very high blood pressure, someone who had a recent brain tumor, who's had major surgery, just a bunch of criteria that we have to live with. And then we have to figure out if somebody got a dose of TPA and got better and then they got worse again, should we give them another dose? And one approach, because after all, if we're just clearing the blood clots, but we're not fixing the underlying problem, aren't they just going to make new blood clots? And so we've been considering or using another type of anticoagulant so that after we clear the clots, we keep them from clotting again. And our hope is that we will be able within 24 hours to see some benefit, either because we can increase the amount of oxygen in their blood or we can decrease the amount of oxygen that we need to give to, and then some secondary outcomes. Did they survive? How long were they in the ICU? Did they make it out of the hospital? Interestingly, the data analysis turns out to be quite complex, and I can't pretend to understand it. But those of you that are interested in statistics could think about how to do these kind of trials. It's what's called a linear mixed model with some estimating equations to try to figure out how many patients we need. And again, the sample size was determined based on some estimates of what it would take to see benefits. And our hope is that within about 30 patients, we would have the ability to predict whether this will be beneficial or not. I think I'll stop there, and I'm happy to take any questions about the trial. But I think you've got a gist, a feeling now for what's involved in trying to figure out how best to do this. So I'm going to stop screen sharing, and I'm happy to take a few questions.
Excellent, thanks. So I will basically bring people off mute who have the questions up. If you have any questions, just the quick tutorial on this, because some people don't know the hand raising. If you click on the participants tab that you should see your name and a little hand raising icon. So if you have a question, click on that and we will get to you. So our first question here is from Robert. Robert, I've taken you off of the mute on my side, and you can do the same on yours and ask your question.
Yes. Hi, Dr. Yaffe. It was a very fascinating talk, and I'm very interested in the mechanism of PI1 in this thrombosis in COVID-19. And what I was wondering about, I understand the way the trial is structured now. I was wondering if you considered any of the PI1 inhibitors. The statins are known to reduce synthesis of PI1 by the endothelium, and there are also these diketopiperazine-based inhibitors as well. It would seem to me you might get as good an effect if you inhibit the PI1 as your current modality.
So I think, Robert, those are both excellent points. And actually, we probably should have considered those. I still think we need to overcome the initial blood clot. But certainly, both of what you're proposing are actually excellent ideas once we get the initial clot cleared, right, to be able to give something to prevent elevations in PI1 levels. Now, we had been leaning towards using anti-IL-6 therapies because our hope was that that might not only affect PI1 levels, but would also reduce all of the other effects that IL-6 has on thrombosis. But I'd be quite interested in hearing more from you offline about other drugs that are available.
Wonderful.
Block PI would be an excellent way to proceed.
Thank you, Dr. Yaffe. I will contact you shortly.
Excellent.
Great. All right, so I'll take a question here from Caleb, and then I have Helen.
Hi, great talk. My question is whether you considered, you mentioned rebolusing with TPA. How about a longer acting clot buster like streptokinase instead of TPA?
So that's also an excellent question. So the beauty of TPA, and the reason that we like TPA over streptokinase, is that streptokinase activates lysis or can, I should say, activate the degradation of fibrinogen and everywhere, systemically in the body. Whereas TPA, because of its Kringle domains, only can activate plasminogen if they're both co-localized on the blood clot itself. Now, an excellent idea, and this is something you guys could consider, could we find a way to encapsulate streptokinase in a nanoparticle that was uniquely targeted only to bind to fibrin clots? And that would accomplish exactly what it is that we would set out to do and would have the advantages of streptokinase itself. And that again is an excellent idea. But at the moment, the reason we've leaned towards TPA is because of its limited ability to activate plasminogen only on the surface of clots. Now there are, as I suspect you're aware, alteplase, which is one of the forms of recombinant TPA, is the one that we've used mostly because it itself has a short half-life. There are alternative forms of TPA, tenecteplase is one of them, where a few mutations have been made that prolongs its half-life. And we jokingly refer to that as zombie TPA because it's TPA that never dies. But my concern about that is that we might see a higher incidence of bleeding complications. So it's a balance between having prolonged clot lysis, which is good, versus increased bleeding risks, which would be bad.
Thank you.
Just a second, I'm scrolling down to find Helen on the list here. Oh, maybe you popped it to the top. Sorry. There we go. Yep, we got Helen there.
mentioned
right.
at the, oh, can you hear me?
Yep, we should be able to hear you now. Thank you.
Very good. All right. You mentioned at the top of your talk that you noticed angiogenesis in the lungs of these patients, and I've also read that elsewhere. I'm wondering, do these new vessels succumb to damage because they have active COVID-19?
That's a great question. So what's strange, at least in the New England Journal paper, about these vessels is the way they form is by an alternative route than what we're typically used to seeing. So as you, I suspect, know, we think that normally blood vessels grow from some type of vascular endothelial stem cell, which then invades in the vicinity around the around where the blood vessel itself is going to form, and then they grow and branch as they grow into that extracellular matrix. What's been described, at least in the New England Journal paper, is this sort of splitting blood vessel, where in fact the blood vessel that itself is clotted then begins to split laterally and extend from there. And so presumably that's already a vessel that's been damaged. So my concern is exactly what you're suggesting, which is that even though there's a response of the body to try to make new blood vessels to find a way around the blockage, that those blood vessels are growing adjacent to a vessel that's already been damaged by the virus. And I suspect that those blood vessels themselves are likely to succumb just as the initial ones from which they're sprouting did.
Interesting. Thank you.
All right, we have a question from Christopher.
Hi, Mike. It's Christopher Hoog. Excellent talk. I had a question that, have you looked?
So I say again, Chris, I think we dropped a little bit of what you said.
So I wondered if you had looked at individuals who might be on other medications that might predispose them to bleeding diatheses. For instance, aspirin, excessive omega-3 supplementation, other things of that sort, whether these patients might be protected in this setting. Of course, if so, you may never see them.
So if you don't know, Chris is a practicing pediatric oncologist, a very, very smart guy, so he knows exactly what he's asking. We've had an interest in this for a while, and there's an ongoing study to look at this. My personal belief, but I don't have any proof for this yet, my personal belief is that patients who are already taking normal anticoagulants for some other reason, antiplatelet medications, aspirin, Coumadin, factor 10A inhibitors, because of some other reason, I think those people are protected from ARDS induced lung failure. I'm working with one of my colleagues, Dr. Gabriel Bratt, who's an expert on big data, and we're trying to look at that data now to see if we can propensity match, because you have to match, the problem is those patients are in a different class than average patients, because they already must have some medical issues if they're taking anticoagulants. So it's hard, yet it takes some work to balance, frailty match those patients against what would seem to be otherwise good controls. But we're looking intently at that question, because as I said, I do think that patients who are on natural anticoagulants may be protected from COVID-induced ARDS. I don't think you're protected from the virus, but I think you're less likely to develop ARDS if thrombosis, it really does play a major role, as I think it does.
And this reminds me of a talk I attended by Dr. Judah Folkman a number of years ago, where he discussed endostatin and the hypothesis that this would prevent inflammatory diseases. In particular, the Kawasaki-like phenomenon that was happening in children, he predicted that that would be absent in individuals with Down syndrome because of the extra copy of endostatin, which is located on chromosome 21. So he asked us to refer any patients with Down syndrome or cancer, with cancer or Kawasaki's disease to him. And I guess I would also inquire whether anyone, I know it's a smaller number of patients, but individuals with Down syndrome may also have a different propensity to develop COVID-related lung disease.
That's an excellent question. I had not thought to look at that, but now that you say it, we definitely will. One of the things that we know is one of the things that's been so interesting about this disease is that the the white blood cells that seem to be involved in this disease are absolutely fascinating. So in the circulation, as you know, and perhaps many of the audience know, the profile of white blood cells is very different than what you see in a typical viral illness. In a typical viral illness, in the circulation, you find a lot of lymphocytes. In fact, there are very few lymphocytes in the circulation in COVID-19. And instead, there's a tremendous increase in the number of neutrophils and monocytes that will become tissue macrophages, part of this innate immune response. And one of the things that our lab had discovered in a different setting in trauma was that the enzymes that neutrophils release, which are obviously being released at high levels in the lungs of COVID-19 patients, one of the things they do is they degrade plasminogen into something called angiostatin, which blocks blood vessel formation. But the other thing that angiostatin does is it competes with plasminogen for activation. So our argument is if you have a lot of inflammation already, that will also contribute to your inability to break down clots because you're already degrading the substrate that you need to lyse the clots. So I think it's a combination of a variety of normally protective effects that have just gotten out of control from these positive and negative feedback loops that were just never designed to handle a virus as deadly as COVID-19.
Thanks, great.
So we don't have any other hands up at the moment. I give people a couple minutes here to think through additional questions. Kind of along that same line, and this obviously an area that's definitely outside of my background expertise, but do you see anything associated with the age relationship that we often see with people having, the more complicated respiratory failure?
So what's been really interesting to me is the types of people who we see with respiratory failure. So obviously the people that are young, children, really don't present with respiratory failure. They do sometimes present with endothelial injury, but they have not presented very frequently with respiratory problems. And I suspect some of that has to do with the fact that their immune systems are still relatively immature. And I think it also is an indication that as you age, we know there's an inflammatory phenotype, where as you age, your body becomes more balanced towards inflammatory responses than anti-inflammatory responses. The other thing that's been remarkable to me has been the link between this and obesity. Many of the patients that we've seen in the ICU are significantly overweight. And we also know that metabolic syndrome and obesity are also associated with this pro-inflammatory phenotype. So clearly there's some connection between this obesity and age and inflammation that has not really been well elucidated. The other thing I would just encourage everyone to think about is, we've been studying, the scientific community has been studying blood clotting for 100 years. You would think that something as simple as how your blood clots would have been well worked out 50 years ago. But we are constantly discovering how incredibly complicated the blood clotting process is. And we are constantly discovering how little we actually understand about how blood clots in vivo. And that's because we don't to be able to actually blood clotting in real time in the body. So if people want to think of clever approaches to be able to monitor in real time in an animal even how clots are forming and lysing, a huge need for that, because all the work we do in vitro is not cluing us in. And the proof of that is the fact that the lab tests that we rely on for measuring blood clotting are telling us that these patients are slow to clot and are having problems clotting, when in fact, clinically, we know it's exactly the opposite. It's not delay in clotting, it's accelerated clotting. So clearly we're missing a lot.
Yeah, it's very interesting. We have actually one group through the biomaker space that is looking at sensing and detecting clotting. It's a device-based approach, so I think you'd have a little bit more of an issue and challenge here, but perhaps some good discussions and thought between us.
Steve Tannenbaum, I see you have a question.
All right, Steve, you should be able to unmute yourself now.
Ask away, Steve. Steve, I can't hear you. Maybe type your question in the chat. I can't hear you, Steve. Justin, it looks like somebody else is also asking a question.
Yeah, I'll put Steve on mute for a second. Steve, if you want to try typing into the chat window, we can read that one for you. If you can get to the chat across the bottom and type in there, you get that to any of the panelists, we'll ask. So we'll put you on mute for a second. We'll take the question from Sean and come back to your question.
Hi, Mike. I was wondering if we've heard about other epithelial sites for the infection, maybe in GI. And I wondered if the presence in the lungs of this sort of interaction, or the co-presence of the clots and the virus, is that due to the site of viral replication, or is it just that the lungs are a more critical system, or the physical parameters there? What's causing that, or are there there's some case reports of clotting elsewhere in the body, but maybe that's just less important?
Again, terrific questions. So here's what I can tell you that we know. So why the lungs? Is there some tissue tropism to this virus that makes it, is there something special about the lungs? So ACE2, as you know, is the one of the receptors that the virus uses for entry. But we know that there's high levels of ACE2 all along in many tissues, not just the lungs. And there is viral shedding, as I suspect you know, throughout the GI tract, although it's not, we think, as dominant as in the respiratory tract. So I think a lot of it is probably just the fact that the lungs are the viral route of entry and the initial site of viral propagation. Now, what's been interesting, as you also allude to, is that two other hallmarks besides the respiratory failure, particularly in younger people, have been a high incidence of strokes and a high incidence of cardiac complications, including arrhythmias and heart attacks. The strokes definitely seem to be clearly related to blood clots forming in the cerebral vasculature, either in the carotid arteries or in the cerebral vasculature once you enter the skull. The heart attacks are more complicated, and the arrhythmias, and that's because it's unclear how much of what's happening in the heart is endothelial damage versus myocardial damage. That is the heart muscle cells themselves or the endothelial cells. And the initial description was based on the incidence of arrhythmias and other things, that it was really damage to the myocardial cells. But most of the autopsy reports that are coming out aren't finding a lot of evidence of myocardial cell damage from the virus, and are more consistent with damage to the endothelium of the coronary arteries and the branches of the coronaries that supply the different portions of the heart. Now in kids, they're getting something interesting. They're getting a variation to some extent of Kawasaki's disease, which is what Chris Hoog was alluding to. And that is an aneurysmal disease that's also caused, we think, by viral infection of the endothelial cells of the blood vessels of the heart. So I think, to sort of condense my answer, I think the reason we see it so much in the lungs is because that's the initial viral route of entry and it's where the virus gains a toehold. But I think the endothelial damage itself is widespread. Much of it, and renal failure. That's the other thing that's really been a hallmark. We see a lot of people, particularly after respiratory failure, get renal failure. And that may be related to endothelial injury as well. Or it may be related to the fact that in other forms of ARDS, when people get lung failure, we try to reduce the amount of fluids we give them to prevent fluid from accumulating in the lungs. And we may be causing kidney failure by running these patients too dry. So some of that may just be iatrogenic that we're doing in our effort to try to limit the lung injury.
All right, haven't seen anything from Steve yet. Is there anybody else out there with questions? We'll get Steve here.
I can see Steve's trying to reach because the box lights up around his name, but I just can't hear anything.
I wonder if maybe it's a closeness to a microphone location.
Steve, you could check preferences and then it'll ask you audio preferences and you can make sure that your microphone is working.
Well, Mike, I'll ask you while I got a minute. I think everybody's wondering how you manage to stay up with the clinical work, with research work, and everything all at once. It must be completely exhausting.
The easy answer to that is I don't do any of it very well. You know, in some ways, the COVID crisis has been a real breakthrough for me, because the biggest challenge has been much of our laboratory work deals with cell injury in the setting of cancer and cancer chemotherapy. And yet my clinical work was really in the intensive care unit. And trying to balance those two has been challenging. The most remarkable thing about COVID-19 was it really connected all of our basic science work on cell injury and innate immunity and blood clotting and cell survival and death with what I take care of in the ICU. And in that sense, it's really been very life-giving. And I guess if I could give a piece of personal advice to myself, I would have told myself 30 years ago, try to tie your research work very closely to your clinical work. And that makes life really much better. So moving forward, I think this is a real opportunity. And for those people who are at various stages in your career, I would say if you want to do a bunch of things because you're like me and you're interested in everything, and you want to do material science and you want to do biochemistry and you want to do clinical work and you want to, I don't know, scuba dive or whatever, find some way to link everything together that's clear and coherent. And then things build on themselves instead of compete for time. So if I could give myself advice 30 years ago, that's what I would have told myself.
That sounds like pretty good advice. I think one that most of us at MIT could take and listen to.
My advisee, Susan Kong, says less than 3. She says, do less than 3 things at once.
All right. Well, it looks like we're still having trouble not getting Steve's question through. So since we are pushing up right up on the 1 o'clock time, and I want to make sure we're respecting everybody's time, I think we'll need to draw to a close at this point.
Thank you all for listening in, and feel free to email me any questions you have, or if you're working on something related to blood clotting in vivo or innate immunity or how cell damage immunity and blood clotting interact, I would love to chat with you about it and love to help out if I can. Thank you very much.
And thank you. I'm sure I speak for all the attendees in saying it was an excellent talk, and we appreciate your time. So thank you, and thank you everybody for attending. We'll see you at the next session in about 2 weeks.